Hello and welcome. I'm Sam Califer, your host for Vision, the show where we talk all about the College of Arts and Sciences as well as its faculty and students. Today, I'm joined by Dr. Jeff Gout, an assistant professor in the Department of Biological Sciences. Thanks so much for being here.
Thank you for having me on the podcast.
Yeah, I'm excited to have you on and talk about the work you do. But first, I was hoping you could just tell me a bit about yourself and your journey to Mississippi State.
Okay. Well, as you might have already picked up from my very slight accent, it was a long journey. Grew up in France, did all my studies all the way to the PhD in France, and moved to the US to do my postdoc, which is pretty standard thing to do. Did not really know I would stay in the US for a long time. Thought I would be here for 2 years, 3 years, go back home. And I kind of liked it here. And after a couple of years of postdoc in Indiana and then Arizona, I had a job offer that was very interesting for me here at Mississippi State. So I joined the Bulldog family in 2018.
That's great to hear. Yeah, so interesting to hear how you made it all the way here from France. But we're happy to have you here and all of the expertise that you bring. And so as a faculty member in the sciences, you have your own lab, the Gout lab. So what kind of research do you do there?
Yes, so it's in biological sciences, and sometimes you might picture, biologists wearing the lab coat and using pipettes, test tubes, things like that. The tool I use mostly is a computer. I do computational biology, even though we also do use bench work in the lab. My main work is to study genome sequences using big computers. And I also maintain one of these big shared computers for the department, which other labs use. And I'm very interested in what happens when things go wrong in cells, especially when things go wrong during the replication of DNA. For those who remember their Bio 101, they produce mutations which are important for evolution. So what are the consequences of these mutations? What are the consequences of other mistakes that happen during processes in cells? And we work with different organisms. I share a lab space with Dr. Dapper, so we also share resources, work both on nematodes, these tiny little worms, and also yeast, very useful organism. Yeast give us bread, wine, beer.
That's right.
Very important for us.
That's right.
And I have a student right now, Istilan, who is also working in collaboration with Dr. Thornton in the department to work on Streptococcus, which is an important pathogenic bacteria. So we work on a bunch of different organisms, study their sequence, study specifically a process called transcription, for those who remember, and the errors that happen during this process, what are the consequences of these errors.
Sure.
And also I have a lot of collaborations with a lab in California where we work on these things in humans.
Yeah, and it's interesting you say that about the lab coats and the pipettes, because that is the image I have. But I think now in our age, using the computer is super common in biology, and it's something people might not know about. So how do you use a computer for the sequencing and stuff? How do you use that in your work?
Yeah, it's a tough one. For me, it's kind of natural because before studying biology, I had a first degree in computer science. And we use it in a way that might be very different from what people are used to. We typically use programs that you don't even use a mouse to click on things. These are all very highly efficient ways of using computers where everything is done by just typing commands in like black or green screen, whichever way you want to configure it. And the computer itself that we use is actually not even in the building where we're working. We just offload all this work to a really big computer, which is stored in a server room in a different building in Allen Hall.
Sure.
So it's really a world of its own, not very easy to describe without going through a whole class.
Sure.
But if our students interested, I do teach an applied computational biology class, which walks you through how to do this type of things, even for students who don't have any background in computer science.
Sure. Yeah, you have like these huge data sets maybe that you run through the computer and the computing power. It's like, it's not something you could do by hand. And so I think that's something people don't realize about biological science.
Yes,
nowadays.
and a lot of these things, there are even things you couldn't even do on your laptop.
Right.
Your laptop will you will hear the fan spinning, and your laptop will go in full crisis mode just because it will run out of memory, and it will take years to crunch all the data that we are capable of generating these days, mostly because sequencing machines are so powerful, they spit out so much data.
Right.
So really we have these big computers that are capable of doing the same work as what 1,000 small computers will do if we were to put them together.
Sure. And the way you described your work was interesting. You said you're interested in what goes wrong or what can happen when these mutations happen. So, yeah, I mean, what are you researching and what would you say you're trying to discover?
So initially I was really interested in just the mechanisms, try to understand why, and not necessarily the applications. And mostly through this collaboration with Dr. Mark Vermust, who is at University of Southern California, we've started being really interested in what could be the consequences of some of these transcription errors. So these are a little bit different than mutations, but we're not going to get into the details today. And we're mostly interested in trying to understand whether these errors could play a role in some of the diseases that develop later in humans.
Right.
So typically, neurodegenerative diseases, such as Alzheimer's disease. These are things that tend to develop when we get older. And it's still a mystery what is the reason why these diseases develop. We don't really understand. We can see what's happening inside the cells, but we don't really know why things start to go bad. And we're interested in looking at whether an accumulation of these errors over time could lead to some of these diseases. So that's one of the exciting areas that we are taking the research.
Sure, yeah, that is so interesting. And yeah, I mean, like you said in Biology 101, I mean, you learn that your cells are always replicating, your DNA is always replicating itself, and so anything could go wrong in the transcription, and you're kind of just looking into what kind of consequences that creates.
Yes, and these are very highly accurate mechanisms. So one of the difficulties is to develop methods that allow us to quantify how frequently these errors happen. So it's kind of the same as reading a book and finding the typo. If you have a typo for every 1 million page,
right,
something like that.
Yeah.
You need to be very accurate at finding these things. So we need to develop methods that are extremely accurate at finding these.
That is so interesting. Yeah, I think, and the way that science works, you might discover something that years later someone has that information and they can make a big discovery too. So it's so interesting how science builds on itself, and maybe you can have that big discovery and help someone else have their discovery. So interesting to hear about your work. And another reason I wanted to have you on is because I know that you have a really big interest in photography. You've got your own website, Earth and Sky Photography, and some of your photos have been featured on the NASA's website for the astronomy picture of the day. And so I just thought that was really interesting, something people might not think about. So can you tell me a bit about your passion for photography?
Yes, sure. So outside of the lab, when I go home, I do have a couple of hobbies, and astronomy and photography is really a big one. Takes up a lot of my time outside of work. It's something I started very early. I think I was 10 years old when my grandfather bought me my first tiny little telescope. And since then, I've just been fascinated by looking at the sky. And there are limitations to what our eyes can see, but as soon as you hook up a camera to a telescope, you can start seeing more and more things. What is also really interesting for me is that we can even do science with just a simple backyard telescope and a camera. So that's also something I like to do. I do post nice pictures on the website, which I don't update the website very often. It's mostly just a way for me to archive my pictures. Right. But I do like also to use the telescope to do science, to learn more about different things like stars with variable brightness. You might have heard in the news that there's going to be a new star in the sky soon. There's
yes.
something called a nova,
Okay,
which this specific star, which goes through a cycle of 80 years, and every roughly 80 years, it all of a sudden becomes, I don't remember the exact numbers, but something like 1 million times brighter than normal, and it's going to become visible to the naked eye. So I do have a telescope which I set up every night that the skies are clear, and this telescope just automatically takes pictures of this and tracks this star and sends me an alert if anything abnormal is happening while I'm in bed.
Yeah. I think that's so cool. I mean, the same way that you're just interested in biological sciences, you're interested in astronomy and photography, and that's something that you do. And I think a lot of times, like students or people who don't know professors very well, like put you in a box. So they wouldn't think that a biological sciences person is also interested in astronomy, but it's just a love of knowledge and learning, and so I thought that was a really interesting way that you kind of dabble in another kind of science. And so I was wondering, has there ever been a moment where you realized that this interest in photography and your work as a biological sciences, maybe they have been impacting each other in a way you hadn't thought about?
Yeah, so maybe more the astronomy side of things more than photography in itself. But one thing that I really enjoyed with teaching here at Mississippi State is we have the opportunity to teach, to develop a new class and do something that's called special topics. So we can try, see if it works. And I had the opportunity to develop a new class called astrobiology, which is interested in searching for the origins of life on Earth and asking the question whether there are forms of life elsewhere in the universe. That was a really good way for me to bring both of my patients together. We go through different hypotheses about how life may have started on Earth, which is still a mystery. No one knows. And that's the first thing I tell the students. There's going to be lots of questions, not very many answers during the class. But we also learn things like try to understand our place in the universe, how big the universe is, and also how did humans measure the universe? How do we know how far away we are from the sun? How do we know we're in a galaxy, how big it is? How do we know how far away the other galaxies are? And by that, I don't mean just like looking at Wikipedia and getting the answer. Like what are the different methods that people have come up with to find this answer. So it's a pretty exciting class. I enjoyed it.
Yeah, I think that's a great example of, all knowledge is connected and you're just interested in inquiring, and that's a great way that those two passions are put together. So thank you so much for joining me today.
Well, thanks again for having me. That was great.
Yeah, it was great. And thank you all for tuning in to this episode of Vision. We'll see you next time.